In this video, we will discuss about the Glycocalyx, which is even called as armor of the cancer cells. So Glycocalyx is not only found in the cancer or tumor cells; they are also found in the normal healthy cells. But the Glycocalyx composition that are found in the healthy cells is completely different from the compositions that are found in the cancerous cells. So in briefly, we can see the image. This is the image for the glycolycs that are found around the cancer cells. In the healthy cells, this glycolycs helps, or we can say, is the first point of cell-to-cell interaction, and it contains the various receptors uh, that are able to trigger intercellular signaling that are essential, or that are the essential function of cell-to-cell interactions. So in this case, we will not cover the healthy interaction of between two adjacent cells. However, in this video we will cover how this glycolycs act as a armor of the cancerous cells. So in this case, we can see that this glycolycs of the tumor cells is too densely crowded, and this densely crowded is one of the reason why it acts as a armor of the cancer cells. So this densely crowded region of the glycolycs prevent the surveillance of the immune cells, so that the immune cells will not be able to come and interact with the receptors that are present on the tumor cells, or the specific antigens that are present on the tumor cells, as this glycolycs provide a physical barrier to the immune cells. And simultaneously, what happens is that there is a upregulation of the upregulation of the upregulation upregulation of the aberrant glycosylation. For examples, this sialic acid glycosylation get increased, that and one of the specific sialic acid related glycosylation are Siglec-7 and Siglec-9, and this Siglec-7 and Siglec-9 is also known to be also known as this immune signal inhibitor response. So in other words, these Siglec-9 and Siglec-7 are, in general terms, are also called as don't eat me signals. So they will give the signals to these immune surveillance cells that to deactivate themselves. For example, it will deactivate this phagocytosis and antigen presentation on this we can say dendritic cells, mature dendritic cells, or the T cells. And simultaneously, simultaneously here it is represented here is that the macrophages functioning against these tumor cells will also get ineffective, and it will also trigger we can say it will also trigger the suppression of these T cells, which is equivalent to the don't eat me signal by the Siglec-7 and Siglec-9. And there is a some of the receptors that are found, or that are essential to trigger the immune response, or to trigger the immune response when they interact with the T cells and the dendritic cells, and these are also been inhibited by this syndican that is shown here. And some of the recent publications has provided us studies about the O-glycan interaction in this glycolycs formation in the tumor cells. So if the O-glycans will get triggered, this will reduce the thickness of this glycolycs, and that will make the immune cells become more effective against the tumor cells. And simultaneously, if overexpression of some of the genes that are that leads to the extension of the O-glycosylation, will cause the increase in the thickness of glycolycs, and that will reduce the efficiency of this immune cells against those tumor cells. So that study shows that the glycosylation plays a role in the aberrant glycosylation plays a role in the glycolycs armor formation. And apart from this O-glycosylation, they also shown that one of the studies also shown that this galactins galactins also act as galactins in the tumor cells also act as an immune suppressor, and it is also somewhere correlated with this glycosamine with this glycolex glycolex thickness. And simultaneously this glycosamine glycan function glycosamine glycan will be physically shielded through with the help of this extended aberrant glycosylation that we can see here. It will prevent the diffusion of these antibodies or this surveillance cells to come and interact with the antigens that are present on the tumor cell surface. For example, this CD44 is shielded by this extended glycosylation barrier. So I hope you have understood how this glycolex act as a armor for the cancer cells. If you need to understand more about this glycolex and its compositional analysis, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.